Our next talk is by Sylvain Baillet from Montreal Neurological Institute. And he is a physicist and graduated from the University of Paris with a PhD in 1998. Then he moved to University of Southern California as a postdoc with Richard Leahy, and did some very influential papers during that time, and a little bit after that, together with John Mosher, and also I think Dimitrios was already involved at that time. Were you?
Actually, no.
Not yet. So you were a bit later. Anyhow, after his postdoc in USC, he joined CNRS in France as tenured research scientist, and worked from 2005 to 2008 as the head of the brain imaging group at the Paris MIG Center. But then he decided that it's time to do something new, and moved to Milwaukee to start a new MEG facility there. And then he is now moving slowly, maybe back towards Europe, because he last fall moved to Montreal to start a new MEG center there, and I am sure there will be many exciting possibilities to study how both the healthy and diseased brains behave at rest, even though I doubt whether there's such thing as rest for our brains. Okay, please.
Thank you, Matti. Thank you so much. Thank you, Dimitrios and David, for starting the clap. Yeah. Well, thank you, the organizers. Thank you, Bob. Thank you, Charles and Dimitrios, for the very kind invitation. I'm very thrilled to be here and share some of my enthusiasm for the technology. And I wish the MEG Center at MIT the great success it deserves. So this is a word cloud that we designed for a training workshop we organized in November back at McGill. And I guess it reflects pretty well the impression a newcomer to the technology has. So there is MEG at the center, loads of data. We're talking about imaging, detection of signal amongst noise, neuroimaging, registration of subjects with MRI. That's a lot of things and tricks you need to learn. And so we'll try to take you from A to Z with examples today in the context of the resting brain. The good thing, though, is that in the middle of this word cloud, there is always free coffee. So that's good, right? That's what keeps me going, I guess. And before I'm running out of time, I'm really grateful to my collaborators in Paris, Milwaukee, and now Montreal. And Beth Bock is our system manager. François Tadel is the core software developer. I will have a word about his work a bit later. But for those of you who have signed up for the workshop tomorrow for Brainstorm, so François is the key person to talk to, and then of course collaborators in Montreal and elsewhere. And I'm very grateful for Richard Leahy, Matti Amalainen, and John Mosher for a long-term collaboration. But today, most of the work I will be introducing is from the project of Esther Florin. She's a German-trained physicist specializing in time series analysis. So a little bit of tourism. You have heard about the MNI, I guess. It's the first integrated clinical and basic neuroscience program, I think, worldwide back in the 30s. Now there is a new extension to the old and venerable Neuro, as we call it, building. And this is where the MEG is sitting. And it's been open since about March and officially September last year, 2011. So it's an MEG. And for, again, all the results I'm going to show you today have been processed using the toolbox we will be featuring tomorrow during the workshop, which is called Brainstorm. Okay, so as promised, I'm going to talk to you about MEG and the resting brain. But before, I would like to segue a little bit and put the brain at work, because I'm going to tell you the story of how we came up basically looking at the brain from a resting or ill-defined, if you will, state perspective, and therefore deviate a little bit from the tradition of looking at event-related responses in functional mapping, if you will. So I'm going to be showing you for the next 2 or 3 minutes a few examples that I think represent pretty well what you can do with MEG source imaging and complements in some aspects what we've been attending to until now through this exciting workshop. So, for instance, I think Rita Hari has very well explained that designing a MEG study gives you a lot of new dimensions when you come from an imaging background with fMRI or PET expertise. And the first thing you have is, of course, the temporal dimension, but it's not so much not only in the time series analysis you get eventually, it's in the very design of your paradigm. And you can really push the limits of the temporal resolution and play with that a little bit or a lot so that you can really give yourselves the maximum amount of chances to get the best signal-to-noise ratio in your analysis. So there is a very well-known protocol in animal electrophysiology and to some extent in human electrophysiology as well. This is the rapid serial visual presentation, RSVP paradigm. And it's actually very simple. What you do, for instance, you can do that with objects, but it works really well with faces. You present a series of faces that are unknown to the subject, and eventually, every once in a while, you're going to introduce the face of somebody these persons know. And this is going to be the target because the subject or the participant will have to press a button. But the thing is that because this is rapid, we're going to present each face actually at a very quick and fast rate, every 50 milliseconds or so. So it's extremely fast. So basically what you're going to get is a visual response from the visual cortex and maybe some of the rest of the brain that pertains to face processing. But in the middle of that, you have a target, and therefore the subject is going to press a button, so there is a task performance that is engaged, but the flow is keeping up. And you may question what you obtain for targets versus unknown faces in terms of contrasting the responses, even though they are captured in the same very quick flow. So is the brain stopping for a little while, looking at things in its environment, or is it keeping with the flow and it doesn't matter? So what happens basically, and this is a little movie showing you what's happening here, and it's going too fast, of course, so I have to pause it for a little while. So this is an inflated view, obviously, of a brain. And this is during the first 500 milliseconds following the presentation of the target face. But then you have more faces coming up. So you really need to compute the event-related average, for instance, of the response to the target face minus the event-related average to the control, if you will, or control face. And what you get is not so much activity in the visual cortex, the primary visual regions, because they get canceled out by the subtraction. But the very first strong signal you're obtaining is actually in the ventral part of the temporal lobe, like you've seen just before. And then it spreads very rapidly, like here. So not too far from the fusiform face area. And then you get propagation essentially over the ventral stream, but also to some extent over the dorsal pathway. And then this is a medial view of the brain. And this is the 300 millisecond response to typically what an oddball paradigm generates. And whenever there is a target among distractors, you get a very strong response at 300 milliseconds while the rest of the story is going on. And basically, the brain is busy processing that target face. It makes sense. It's very intuitive. And that gives rise to some kind of buffer time where you cannot have the brain perform another task, and that pertains to the attentional blink paradigm. And there's been a lot of study about the attentional blink, but I can direct to some studies we've been performing with Stan Dehaan's group recently. So very simple design, although you push really the limits of the brain, I guess, in terms of processing, and just by encoding and generating a series of very fast presented series of pictures. If I switch to something a bit even simpler than design, what we tend to forget is that the MEG scanner is actually very versatile in a way. You can operate supine, as in a PET or MRI scanner, but you can also operate vertically in seated position. And also it's very quiet, as you know, so you can really run, like we've seen this morning with Pat Cool, studies in a very friendly environment, especially for children and patients. So that collaboration with Patrice Senot back in Paris was very interesting because it was challenging in the very design of the experiment. Patrice was very interested in the visual processes involved in hand-eye coordination and especially in object interception. And what he's been doing is actually design a fairly ecological paradigm whereby the subjects were basically asked to catch tennis balls that were falling in front of them irregularly. So they could not predict when one would fall. It's a very stressful situation. But actually the pace was relatively slow. And at time 0, the ball is released and enters the visual field of the subject. And by 350, 400 milliseconds, the ball is caught if the subject, make a grasping movement. So what I'm going to show you is actually like a synoptic view of the slow processes that builds up in the brain as the hand-eye coordination is programmed and eventually realized. So this is the time scale here in milliseconds. The ball is released at time 0, and then it is caught on average at 400 milliseconds. The white line is just here as a guideline, if you will. This is the averaged instantaneous brain activity. So we take each and every source on the brain and just compute the amplitude in a fairly slow and narrow band below 5 hertz. And we just look at the slow components that build up in the brain. So this is the global brain activity, if you will, that slowly builds up with the task. So the little red circle is going to move from baseline and along this roller coaster. And these are two views of the same brain, a little bit inflated, so that we see deeper into the folds. So you see the little time cursor moving in. Time 0, the ball is released. Nothing gets above threshold, except by time 50-ish millisecond, where you see or cannot see from here some primary visual response to the ball entering the visual field. But you see also, and in that subject it's more unilateral than bilateral, you see some extrastriate response, maybe not too far from the human equivalent of empty, which is an area involved in motion processing in the visual field. So by 50 milliseconds. And then the activity spreads out and travels essentially over the dorsal pathway before it reaches the motor or sensorimotor cortex contralateral to the hand that's going to be catching the ball. And so you have to remember that the ball is only halfway through. It's still traveling and still falling. But to some respect, also because of the repetition of the task, the problem is solved by the sensorimotor cortex within 200 milliseconds or so. And then it goes back to a plateau. And again, we don't see very clearly, but if we had a medial view of the brain, there would be essentially V1 plus activity over the medial wall or interhemispheric walls of the brain. You see that there is a plateau that is really above baseline here. So probably just checking that everything is going according to the plan. And then it's only by 350, 400 milliseconds that the sensorimotor cortex is basically triggered again directly without a traveling wave, or at least we cannot detect it if it's here. Apparently the motor program that has been encoded is released and executed at the right time. So I guess this little movie, and also I forgot to mention, you see some ipsilateral cerebellum activity. So there's been always this debate about detecting or not cerebellar or subcortical activity with MEG, and I think the answer is yes if you give yourself the right design of your experiment. Okay, so this little movie, I've been using probably 3 minutes of my precious time just to explain 400 milliseconds in the life of that brain for a very simple task, or relatively simple task. And I think that's all the beauty, or some of the beauty, of MEG, that where maybe the spatial resolution when we apply this kind of straightforward techniques is not so great, but great enough, if I may say so, so that you are able to really look at the whole brain as a map, basically, resolved in time, obviously. So talking about spatial resolution, we wanted to push these limits a little bit, and I'm glad that Rita mentioned frequency tagging this morning. And we've been using it in maybe even a more straightforward manner in that study that my former PhD student, Benoît Cotreau, performed. And basically, we took advantage of that temporal resolution of the processes in the brain responding to a stimulus. So if you flicker, like we saw this morning, a stimulus at a frequency F1, you get a response from the brain regions that primarily are engaged by the stimulus, maybe at F1, but in the case of visual processing, with the kind of stimulation objects we used, the strongest activity was at the first harmonic of the stimulation. So here we use some stimulation frequency at about 8 Hertz, about the alpha range, so 7.5 Hertz. And we did that, and the goal was to basically derive a step-by-step procedure that would basically help us obtain a very crude retinotopic map of the visual areas. The goal was not so much to do as good as, or probably not as much better than what fMRI does, but we wanted to have this retinotopic map so that later on we would be able to measure delays within these circuits that are basically engaged while responding to flickering stimuli. So I'm going to show you the data later. So basically, when you stimulate with the blue dot here, you expect to get, of course, the contralateral. Sorry, the green dot is going to be the contralateral response over the primary visual cortex, etc. The primary meridian, horizontal meridian, will be responding or generating responses over the calcarine fissure, etc. So that's what we expect. And when you look at the traces, actually, there is not such a great response at 7.5 Hertz, like I was saying. This is obviously a topography of the power computed over a few seconds and a few trials, and just to look at the quantity of response from the brain. So not so much at the stimulation frequency. At the first harmonic, you get a very strong and nice response. And that response actually is, as predicted, if you look at the topography over the sensors, even though it's very crude, you get a good sense that it's traveling with the position of the stimulation in the visual field. And then if you look through time, you look at the dynamics of that steady state response. So the stimulus starts at about time 0, and then there is a little transition at 7.5 Hertz, which is the actual stimulation frequency. So that's interesting, but we haven't looked at this thing from too close. We were more interested in the steady state part of the things, and you see a very nice response that basically lasts until you stop the stimulation. So then we applied the basic, if you will, methods that very standard retinotopic mapping does in fMRI, and actually we've seen some of our subjects in fMRI as well using the same paradigm. And what you get in fMRI, this is an average across 3 subjects, I think. You get the expected distribution. Again, very simple, but it's good to go step by step, just to validate every step of the way. So fMRI is showing us the expected contralateral symmetry of the primary responses in response to those stimuli. And when you look at the MEG, qualitatively, it's very similar. So you get this distribution of the primary response, which are the preferred response to the preferred stimulus. So again, it's not so much a breakthrough to basically replicate what fMRI can do. But then what's interesting is that you can encode not the responses in terms of amplitude, but this time you can investigate whether the regions you see responding significantly respond with a delay due to either propagation along the pathways or to local computation. And this is basically what the colors are representing here, where basically you see like a gradient, a posterior-anterior gradient from the dark blue regions that are reference for the phase of the ongoing stimulation, response of 15 Hertz. And you see that the colors will turn from blue to red, where we can actually measure delays between the ongoing steady state responses at that very frequency. So this is just a proof of concept that basic retinotopic mapping is possible. It can probably be much more improved, maybe by using multiplexing with a different stimulation frequency in a single shot. But more importantly, you can access delays that might be meaningful to your study in terms of measuring propagation or computational time. And then what's also very interesting in MEG, and that's not so common when you think about it in the neuroimaging palette, you can really use the same instrument and almost the same methods to map brain function, like I've just shown before, and dysfunction. Meaning that you have electrophysiological markers or manifestations that you can pick up with MEG, like you do with EEG, But again, the modeling of the sources is more straightforward because, again, the skull and the different tissues of the brain don't distort the waves too much. So again, to put the story in perspective, we're starting looking at resting epileptic brain activity. And basically what happens, the standard way of proceeding is that you get loads and loads of time series like we've seen in the previous talks. And what a neurologist would do is mark, or an electrophysiologist would mark paroxystic events of interest in epilepsy. So it could be spike waves, slow waves, etc. And then basically look at the sensor map and investigate whether these manifestations are reproducible during the session, etc. And eventually we're going to move to the source time series. And this is an example of a 17-year-old patient from the Medical College of Wisconsin. And what we've seen in her case is a very fast propagation from precentral regions towards frontal, prefrontal, and anterior temporal activity, or regions, sorry, during paroxystic interictal activity. And this was very much reproducible and very much in line with the grid implantation that was derived later on in the same patient. So you see the grid electrodes in green, and the red electrodes are where the seizure onset and interictal activity was the most prominent. And when you look at the MEG, so I think Richard Coppola mentioned that we need ways to condense the information in a meaningful way, especially in a clinical setting. And indeed, you don't want to look at gigabytes, literally, of data, especially if you wear a white coat and you are a medical doctor in service. You really want to capture the essence of the examination. So with Dr. Raghavan at the Medical College, we derived many different techniques, trials and errors, if you will, so that we could put the MEG at his clinical service. And this kind of map looks like an activation map, but it's actually an histogram of how many times these regions marked in red would be engaged in paroxystic events. So basically, we compute each and every source everywhere on the brain, and we count how many times the activity or the amplitude of the activity is crossing a threshold, and therefore we count 1 for each and every event and each and every source each time the activity is crossing that threshold locally. And this is a summary picture that basically tells the doctor where the hotspots might be. And in that case, it was working really well, and you see a fairly nice correspondence between the invasive and non-invasive exploration. And by the way, it corresponded retrospectively when we look at the T1-weighted MRIs, it corresponding to some abnormality in the girification of the brain. And the patient has had surgery in 2009, and she's doing fine. But the good thing is that, it's just for illustration purposes, but this is actually real data in that very patient, is that you cannot, of course, perform surgery everywhere with no possible issue, right? No possible outcome. So you don't want to perform surgery in a hotspot that would also be engaged in language processing, sensorimotor activity as well. So this little movie actually is actual data where we got from that person, where we co-registered the functional activity marked in red after median nerve stimulation with the dysfunctional activity marked in blue with interictal spike activity. And this is just to illustrate again that the same technology opens the same, or two windows, if you will, on two different aspects that are very relevant to the clinical decision. All right. But what happened also is that during a MEG recording, as you know very well, time is precious. And you cannot wait that the patient has a seizure or has too many spikes, and etc. Or if you're working with babies, you cannot work for 3 hours that they are really performing, or you get enough trials, etc. So we came up with the idea of looking maybe at some properties, fundamental properties of the resting brain. And that was a little bit before, or maybe in parallel with what was going on in bold MRI with the resting state networks, etc. So the decision we made was basically to look at the spectrum of the MEG and whether it was changing with time or whether it would be stationary, but not at the sensor level, really looking at the source level and basically map how this activity would translate and would be distributed. So if you don't get any paroxystic event in an epileptic brain, or if you get a normal subject, you may want to scan this person for whatever the time you have ahead of you, and then you can cut the runs into segments, but that's a detail. Basically, what you do is compute the power spectrum density, for instance, in the different frequency bands, but at the very sensor level without averaging. Of course, there are no events. So you go and do the thing of transforming the data to brain space. And then you can build up sample statistics, empirical statistics, so average of the different distribution from delta to gamma, standard deviation within a subject, within a group, across groups, of course, or any other kind of sample statistics. And the idea is that whenever we had a patient, we would do exactly this, and we would compute the distribution of, for instance, the delta power in that person's brain, and we would get voxels that would be higher than a threshold here or here. We would do the same with theta. We would do the same with alpha, beta, and gamma, but we had no clue whether these things would be significantly, deviating from normal variance. So that's a big issue in a clinical setting, but that's also a big issue if you are interested, in capturing some structure and some maybe coupling between brain regions or within the same regions between frequencies or across regions and across frequencies, looking at coupling between the spectral activity. So I'm going to try to take you there for the rest of my talk. So to address the part of the story where I was mentioning that, okay, we have this patient, can you tell me whether what you see here is significant? So what we did basically was, z-scoring the activity or standardizing the amplitude within that person's brain, and whatever was above 3 standard deviations, especially into that brain, would be above the threshold. But is it abnormal or is it just normal? Like, for instance, the alpha power is stronger over the parietal regions. That makes sense. It doesn't make that person epileptic. So we came up with the idea, and it's an ongoing process, and I think it's worth joining efforts with other imaging centers. So it's an open invitation to data sharing and grant submission to build a data bank of healthy brain activity, a little bit along the lines of the connectome, and because this is the way neuroscience, or one of the many ways modern neuroscience will be performed these days, especially with large cohorts of patients and controls. So for now, we are building these things gradually, and we have 45 people enrolled so far, but that's a very small number. We need thousands, basically. That's why we need to share data. But for each and every one of these 45 persons, we basically did exactly what I was mentioning. We computed, for instance, here just the average of delta activity during resting state, whatever that meant. But basically, they were not moving, keeping their eyes open, I think, here. But I'm not even sure this is important. We'll see. So this time, the colors represent basically the strongest and most prominent brain areas generating delta activity. And so a lot of anterior and prefrontal activity for delta, also temporal activity. For theta, it's a little bit more frontal, more posterior, and pericentral activity, going very deep, and why not too far from the thalamus for thalamocortical circuitry. Alpha, as expected, is very much over the parieto-occipital regions. Beta, not too surprisingly, is essentially on average across these people and across the sessions over the sensorimotor regions. It's essentially one of the idling rhythms of sensorimotor activity. And then gamma, and we opened up the filters actually very high. All this data is sampled at 2 kilohertz because you never know. I mean, as we look at the recent neuroscience literature, we can find even task-related neural oscillations that go up to 1 kilohertz. So, and today the systems can sample up to 6 or 12 kilohertz in MEG. So it's just a matter of managing large amounts of data. But so far we've been looking at up to 250, 300 hertz. And that's interesting because, okay, we don't learn too much from alpha, beta neither, but it's good to see that it reflects what we were expecting, about the same for theta. But for delta and gamma, what is striking is that it's not very well localized in a way, but it's similar in the projections. So high frequency and low frequency in power, at least from that very crude perspective, on average across 45 people, they tend to demonstrate similarity in mapping. So now the idea, you get that patient, for instance, resting in the MEG scanner, and you want to understand whether this activity over the perisylvian region is a normal deviant or is outside the normative distribution. So the statistics are essentially empirical statistics, and basically you take each and every voxel on the cortical volume or surface, and you just look after you have computed your statistics in different frequency bands or different coupling metrics, for instance. You want to understand whether this patient is sitting at the tail of the distribution or within the full width and maximum boundaries. And you do that for each and every voxel. So we started to look at this approach with some epileptic patients. And basically what you find, it's been helping us a lot tremendously, I should say, in our design of the threshold, because basically the threshold now is objective with respect to the expected deviance from a normal distribution. Normal in the sense of healthy controls. And so in some cases we find very focal, variations in the beta range, in the gamma range, that coexist very well and correlate very well with the mapping that is done in a standard way in the clinic or with grids. But in some patients, we see some cross-frequency interplay, if that is what it is. Like, for instance, everything that is orange in the central region for that patient is an increase or an expected level of delta activity in this region. And in the same patient, at the same time, you get a decrease of beta activity unilaterally. There is nothing on the left hemisphere. And that is abnormal in the sense that it's not expected from the normal distribution. So we don't know what to make out of this. It's just, we are in the process of building a body of knowledge and a body of data to basically investigate that a little deeper. But again, for a clinical neuroscientist, you don't want to show thousands of maps or hundreds of maps in delta, gamma 1, gamma 2, gamma 3, etc. You want to compile something in something that is synoptic and that could compress information effectively. So we end up doing also histograms of hits, meaning that every time there is a hot color here, it means that the voxel has been detected as a deviant from expected normality. And we are in the process of running a group study on epileptic patients, and so far the results are really encouraging. So this is the epileptic brain at rest. But there's been this literature about the anatomy of the resting brain in healthy controls or healthy participants. Starting with PET studies with higher metabolic activity in the frontal and prefrontal regions. But also you all know that probably that with the latest developments of resting state science, if I may say so, essentially with fMRI, there's been default mode networks or resting state networks that are shown here in the very early meta-analysis that were performed and that are regularly refined as we are making progress in the techniques and actually working on a greater body of data. So for instance, the different colors you see here tend to highlight regions, brain regions, that tend to exhibit correlated activity in bold during rest. So you find, again, the default mode networks in orange, the dorsal attentional network in blue that we've seen with Pascal, I think, this morning, and the green frontoparietal network is also very prominent in fMRI. I'm just going to skip that study. Of course, working with MEG, you may ask the question whether there are some dynamics and stationarity in those networks, because they seem to be so reliable and reproducible using BOLD. So how come we cannot find traces of this resting state activity with EEG? What's interesting also on the BOLD, in the BOLD community, is that they're pushing the limits of time resolution. This is a movie. Is it running? Yes. This is a movie from Marcus Raichle he published recently in a very nice review paper about resting state. And this is BOLD activity showing correlation maps that are evolving with time. So every image is possibly a sample taken at every second or so in the course of the BOLD time series. But you see it's not a still picture. It's really a movie. And correlation is actually being updated at a fairly slow rate. But we want to understand whether what you see here or how what you see here can be correlated to different resting states that are well qualified and well categorized in EEG or MEG. Like somebody very excited, very aroused would show very, very rapid fluctuations in its EEG ongoing traces, and that's resting state. Somebody very relaxed would show very strong alpha activity, and that would be resting state. Somebody being a little bit drowsy and falling asleep in the scanner would show very distinct patterns of resting activity, and that's resting states as well. So how do they correlate with the physiology of BOLD, basically? So that's why in the last 10 minutes or so, I would like to show you the ways and a model that would basically take you step by step by trying to predict BOLD time series or pseudo-BOLD time series from MEG source time series. So if BOLD is reflecting some very strong neurophysiological aspects of brain activity, we should be able to predict that from MEG, provided that we think that MEG is capturing a whole lot more in terms of the spectra of brain activity. So I just want to mention that it's an emerging subfield of MEG. There are not too many papers discussing about resting state MEG, but there are a couple of groups with very interesting papers, but with a very data-driven approach. There's not so much modeling in terms of explaining the physiology. And there's a special workshop at Biomag in August in Paris, so don't miss that. So we're going to go back to the spectrum of brain activity, and we're going to try to understand and find our way in that spectrum, in that complexity of the data. And of course, we want to understand what are the frequency bands that tend to correlate the most with BOLD. It's been evoked a little bit today. And there's been quite a few papers now discussing about this correlation between BOLD fluctuations and electrophysiology. There is, for instance, this Schmuhl and Leopold paper showing strong correlation between BOLD fluctuations and gamma range activity in monkeys. There's been, in humans, intracranial electrode recordings where, for instance, if by chance you get an electrode in the posterior cingulate cortex and you ask the subject to perform a task, so here, for instance, a study where they have a reading task starting at 0, you see a very broad decrease in the gamma range, very broad gamma range of activity in the PCC, which correlates well with the idea that there is indeed a decrease of activity during task performance, because this region is actually involved in the default mode network that is prominent during rest. But then it's interesting to see that in most of the time frequency maps that are published in this literature, they don't show what's happening, or they don't show too much what's happening below the alpha range. I found that study, and there are probably more in the literature, showing, I think it's from Leopold's group also, in monkeys. They are looking also at the correlation between BOLD fluctuations and electrophysiology. And what's very striking is not so much that you find also a very strong correlation in gamma, but you have also a very strong correlation up to the alpha level, from delta to alpha. And also, very interestingly, like a gap, but something that is more like an anti-correlation in the beta range, which makes it very interesting too. But that does not explain, when we look at each and every single frequency band on average, we don't see any structure in terms of resting state networks that are well known. Maybe in the beta range, you see a little bit of that bilateral activity Pascal was talking about this morning at rest. But we don't see any resting state or default mode networks. So there must be something else. And that something else, we went to investigate the hypothesis of phase-amplitude coupling. That is a very prominent hypothesis, first derived by Georgi Buzaki, that basically shows that there is a great deal of, again, phase-amplitude coupling between the lower frequency end of the frequency spectrum and the high frequency end of the frequency spectrum of the brain at rest and during task performance. So we've seen a little bit of that this morning with Pascal, but I think Buzaki also evokes the fact that there might be some central drivers where basically like the hippocampus, for instance, and subcortical systems that essentially resonate at the theta-like frequency range. And on the cortex, you get gamma bursts everywhere, but these gamma bursts maybe are evoked by a task or are generated during rest, but they are not generated at each and every moment, regardless of what's happening in the other end of the spectrum. So the hypothesis that they are formulating and that they have demonstrated in animal models so far is that the gamma frequencies are basically conditioned to some specific phase of the theta cycle. So you get basically less gamma burst release on the slope of the theta cycle, but you get more at the peak or at the trough, depending on where you're sitting. And then there's been this very inspiring paper from Bob Knight's group in Berkeley by Ryan Canolti and co-workers, and it's been evoked this morning also, where basically they wanted to test that hypothesis in humans, and they took grid data from patients, and this is one electrode time series that they filtered in the theta range, actually at 4.7 hertz, exactly using wavelet or equivalent time frequency transforms, detected the trough of the phase of that theta, marked it at an event of interest, and basically epoch averaged the raw time series based on that event of interest. And this is the raw data over minutes of recording, but you see it evoked average about that theta cycle event, the theta trough of the phase. And what you see is a very nice theta, a couple of cycles of the theta activity that degrades as we move away from the event of interest. But what's very interesting is that if you time frequency transform the rest of the signal and look at the distribution of power at each and every cycle of the theta, you see a very prominent and phase-locked generation of gamma activity, pretty broad gamma actually, from 40 to about 150 hertz, that is conditioned to the phase of the theta. So indeed, at the trough of the phase, you get a very strong burst of gamma. At the peak of the phase, you get a decrease with respect to baseline. So to my knowledge, that was the very first demonstration in humans of that very hypothesis. And we wanted to take the challenge to basically generate this kind of maps with MEG, non-invasively. For the sake of time, I'm going to skip that. So what we did, and again, this is essentially the work by Esther. To make a long story short, we had up to 6 subjects, and I'm going to show 2 subjects right now on this slide and more to come. We basically had them for 10 minutes in the scanner, and we applied exactly the same methodology as Canolti did, and basically we looked at each and every point in the cortex, and you get the very nice raw data that is average at the trough of the theta cycle, and then you see bursts of very strong gamma activity that is conditioned to the phase of the theta. And if you travel through the brain, and this is just a few examples because you can really travel through the brain, it is something that is maybe very strong in some regions, not so strong elsewhere, but it's actually significant. Esther has computed some statistical inference, and maybe sometimes it's narrower band, but you do get this modulation of gamma burst by the theta cycle. So I think it's very exciting. Then we looked at optimization of that coupling. So because so far we've been taking that 4.7 hertz theta as gold, but it's very unlikely that everybody would be linked or would be tuned and driven at that very theta frequency bin. And that was very interesting to see Pascal's data regarding the fingerprint of the gamma. So maybe there's a fingerprint of the theta as well, because these 2 guys seem to work together. So far we've been looking only at 6 subjects. And this is a pretty busy map, but basically what it shows we've been exploring from very low delta to very high gamma, 48 hertz gamma, as a possible frequency driver of high gamma burst at 80 plus hertz. And because the hypothesis would be, okay, if there is a low frequency driver, why reducing it to theta, even though the theory is saying it's mostly theta up to alpha maybe, that drives the gamma, but let's test for that. So we've looked at the beta and gamma and compute the phase-amplitude coupling on 6 subjects, and this is the average map we're getting. So it's looking essentially blue and green, meaning that the driving force, if you will, the driving phase of gamma bursting are essentially found a little bit in delta, mostly in theta, and a little bit in alpha, in regions maybe that are not too far from what David has shown this morning, but I'm not sure this is significant on such a small group of subjects. And if you look at how it's distributed, it's hard to find a pattern, but if you want to believe in what you see this PCC region, PFC region that seems to be driven more with delta, a little bit of parietal lobules as well, so maybe some default mode network here. But there is no true pattern. But that's already a good news that, according to theory, this experimental data validates the fact that most of the cortex is driven by phase-amplitude coupling up to the delta or high theta range during rest. I think I'm running out of time, really. 2 more minutes, maybe?
The boss now.
Thank you so much. So, so we've been generating or trying to derive a generative model for Ball fluctuations because, again, if we think that this resting state can be revealed, maybe we're looking at MEG only, but maybe one step at a time again. If we transform the MEG data into pseudo-Ball traces and apply exactly the same methodology as in fMRI, then maybe we're going to find the resting state networks. So we took we made the hypothesis that this phase-amplitude coupling phenomenon is really something that is very strong and widely distributed over the cortex, and that the slow fluctuations of these PAC measures, PAC metrics, phase-amplitude metrics, would maybe reveal time-independent properties from one default mode network to the next. And we would like to predict Ball fluctuations from that, from the gamma burst related to the theta cycle. Remember the figure I was showing? That was showing that the theta up to the alpha and the gamma would correlate very much with the Ball fluctuations. So we have to take, this model captures the essence of these multi-spectral components in the Ball fluctuations. So the model is actually very simple. For each time series and at each brain location, you extract the full broad range activity, and then we're going to zoom in here, extract the gamma activity, and take the envelope in red, extract the theta cycles, and then according to the theory, we're going to detect events that are related to the duty cycle of the phase of the theta. And just for the sake of signal-to-noise ratio, we're going to keep only the 95th percentile in terms of gamma amplitude at each theta trough. But I'm not sure this is necessary. But this is how we did so far. And then, conditioned to that event, we release a pseudo or canonical HRF at each and every trough where we have significant gamma release. And we do that for the entire recording, and you end up having releases of HRFs that are modulated a little bit by a model that has been recently developed at the University of Minnesota related to how many, the dynamics of HRF release when you have repeated events like that. I'm skipping that for now. And then you end up having McBall time series for each and every vertex. And I'm going to finish here, showing just a few results. So if you take if you look at these McBall time series and how they vary at a very slow rate, every 15 milliseconds, you see basically it's very, very noisy, not too many patterns, and you see some prefrontal activity coming up, fading away, and here you see very strong PCC and PFC activity, very highly, very strongly correlated in time over a minute or so, and then decrease of that together along the same pattern as the default mode activity. And then you take exactly the same approach as fMRI, so ICA decomposition or seed-based decomposition, and you can extract networks. Here it's only 6 subjects again, so the data could be better, but at least we have a very nice trends of PCC, PFC, inferior parietal lobule connectivity for default mode. Dorsal attentional network is also very much connected here. Visual activity, for instance, over the superior aspect of the visual cortex, we get also bilateral correlation, projection over the mediotemporal activity and middle cingulate activity. Frontoparietal activity also, or networking, if you will, is very strong as well, and mostly unilateral, as it's been found in fMRI. You get sensorimotor activity, and you get some language looking like networks with middle and superior temporal angular gyrus, Broca, motor activity, essentially over the left region. I just want to finish with something I'm very excited about, which is real-time neuroimaging. Because now if we have a good grasp on some markers in the timing of the dynamics of brain activity that might be relevant to healthy or pathological activity, so we have designed an experiment where we provide real-time feedback to the subject. It's been going on in Richard Coppola's lab, and we pushed that with the collaboration of Gustavo Sudre and coworkers. I think he's in the room. So, and what you get is basically this, where you do have a participant, this is Esther Florin, by the way, looking at the screen, and then there is an image that is changing colors, and the colors are indexed on a certain metric of her brain dynamics in a given region. And then in real time, the movie is a bit shaky because it's homemade, but basically you have the stimulus computer here and the analysis computer. You're going to see a bit better in a moment. And this is brain activity that is refreshed every half a second or so. So full-blown MEG activity over the cortex. And therefore, we can target specific brain regions and train the subject to basically perform a certain task that he would learn implicitly. So here, for instance, we were targeting the superior parietal regions and central regions, and we were asking the subject to improve the theta to alpha power ratio. So you don't know how to do that. But if you are providing feedback, and here over 30 sessions, you see a training, a very nice trend here, a very nice, and for one subject, and a very nice and very strong slope of learning curve, if you will, for that second subject over 30 sessions. And this effect maps very well over the targeted brain regions. Thank you so much for your attention. So I guess I'm going to skip the discussion unless you have questions. Thank you so much. And you can update your Facebook status. We have a make page.
Even though we are a little bit late, let's take a couple of questions. Pascal.
So you showed the gamma and high gamma power to be modulated by the theta phase. And I realized that on some of your examples, the phase was positive, the theta phase was negative. I guess that is simply due to the fact that the theta phase is basically arbitrary, right?
Exactly, especially for energy sources, yeah. Yeah.
Would actually be a way, I mean, looking at this would be a way for you to decide, right, on which theta phase. I mean, you should then probably flip the theta phase.
Yes. Yes.
Based on, because it's kind of, you can make a qualitative decision based on the high gamma power, where it's high, and then you can decide which theta phase.
Exactly. Actually, what we're doing is systematically detecting the trough and the peak of the theta and do exactly what you were talking about. And eventually decide on whether what we're capturing here as a sign for the phase is related to the expected phenomena you're talking about, or whether this is an effect of ambiguity in determining the flow of current with MEG. But qualitatively, the effect is there, yes.
So you're collecting a lot of data from a lot of different patients regarding the resting state of the brain. The question is, without defining what that resting state is, what good is the accumulation?
Yeah, I mean, so far we looked at only 6 subjects with the resting, where we have detected this kind of patterns. And for the database we are building, we cannot control, it's going to be hard to control for, the arousal, the state, the anxiety of the subject, but we can control for some factors like the gender, the age, etc. So as the data bank is growing, we're going to factor out and maybe do a factor analysis of these kind of aspects. For the eyes closed, eyes open, you can give directions, but in an MRI scanner, some centers don't give specific directions regarding eyes open, eyes closed, and even maybe some subjects are sleeping during the resting state scan. So it seems to be an intrinsic phenomenon that is very robust and very strong. So I'm not too anxious about gathering a large pool of data where these factors are not really identified. It would be great, but it's going to be hard. I think this is really, it's potentially something that is very strong and again very robust because it's been demonstrated in so many conditions and centers. But it's a bet I'm taking.
All right. We thank the speaker.